Okay, so today we are going to finish up chapter 17 on viruses. I told you guys this chapter was really short. So make sure the video is not going to be too long today, guys. I only have like 15 slides to go through, but make sure you finish up your reading guide. And then I am also going to post another video for today, and it's just on the coronavirus. So a little animation, a little bit of information about the coronavirus. So to start off, we are going to continue, like I said, our talk about viruses. And last time we concentrated more on bacteriophages, and obviously those are viruses that infect bacteria. Today we're going to move over to ones that infect us, animals, okay? So for viruses that infect us, they might be single-stranded, they might be double-stranded, they might be made of DNA, or they might be made of RNA. So there's 4 different variations we have right there. We could have single-stranded RNA, single-stranded DNA, double-stranded DNA, or double-stranded RNA. So the big thing about the animal viruses is going to be what is on its outer envelope, okay? So we're going to be looking at the proteins on the outside. I know you guys hear about the coronavirus, and it has all these spikes and stuff, and that is what allows them to enter our cells. So that's kind of what the envelope is. So that envelope allows it to gain access to our cells. It's almost like it's knocking at the door and our cells are just opening the door and letting it in. In reality, it's just the proteins are matching up between our cell membranes and the envelope of the virus. All right. So what happened with coronavirus is that the proteins on the envelope mutated, and that mutation allowed us to gain access into our cells. Okay, so we're going to talk about retroviruses, things like HIV and AIDS. So that's a retrovirus. Now, retroviruses are going to be made of RNA. So if they're made of RNA, how are they infecting us? Because we're DNA. So if you remember, we go from DNA to RNA through transcription, and then we go from RNA to a protein through translation. So DNA to RNA transcription, and that's the main one we're going to talk about right now. So what these retroviruses can do is they can go through a process called reverse transcriptase. Now transcription, again, DNA to RNA, if we reverse that process, which is what the retrovirus is going to go through, it's going from RNA to DNA. So what happens here is they are able to use the RNA, make it into DNA, and then put that DNA into our genome, and that's how the retrovirus becomes pretty much part of our cells. All right. Now, since they use reverse transcriptase, what does this tell us about RNA? All right. So since it's reverse transcriptase, anytime you guys see this, make sure you know that it's always starting with RNA and not DNA. All right, if it's regular transcription, we know it's starting with DNA. If it's reverse, we know we're starting with RNA. And then also for retroviruses, like 99% of them, pretty much all the ones we discovered so far, are going to be made of RNA, and they're going to be single-stranded. Okay, so that's retroviruses. So our next slide is going to be on proviruses, and if I can move myself out of the way here. So proviruses, these are the ones that really, really stink. And I don't mean smelly. I mean they're wicked. So what can happen here is a provirus. That's when the DNA from the virus, now it might start off as RNA, but whatever. So the DNA gets incorporated into our cells. When that cell reproduces, the next cell that is made from that, so the daughter cells, they are going to have that viral DNA in them already. So that's a provirus. The DNA from the virus is permanently in that cell, and any cell it makes is also going to have that viral genetic material inside of it. So moving on, the thing that really stinks about RNA viruses is when we are dealing with an RNA virus, they are going to have higher mutation rates compared to a DNA virus. And why is this? Well, if you remember, for DNA, we have DNA polymerase. And DNA polymerase, on the back of it, had that little spell checker to make sure all of our A's were bonded to T's and all our G's were bonded to C's. So when a DNA virus goes through any kind of reproduction, it's not a big deal. All right, if there's a mistake, we catch it. Now, if it's an RNA virus, we're going to use RNA polymerase to make a copy of it. Now, the thing with RNA polymerase, sorry about the bell. The thing with RNA polymerase is it does not have a spell checker on the back. So if there is a mistake when we are replicating, that mistake will probably stay with the virus. What do we call that? Well, that's a mutation. All right. So since there's no proofreading thing on RNA polymerase, the RNA molecule or RNA viruses are going to be much more easily mutated than DNA ones, just because there's not that proofreading mechanism there. Okay, so we kind of already went over this one, guys, with the host cell coming in. Only difference in this picture here is that in the middle, you guys can see the provirus, the DNA, is going into our DNA, and then when that cell reproduces, we are going to have all of our other cells with that viral DNA incorporated into it. So here is a picture of HIV, and we have a white blood cell, all right, and it's attacking it. So it can actually, HIV, infect our white blood cells. And if you remember what white blood cells do, they are the ones that are going to fight infection. So if the HIV is taking over our white blood cells, it's literally compromising our immune systems. So our last part on this section is just about how viruses came to be. And we don't know everything about where the first virus came from and how it actually came to infect our cells. But here's what we do know. We know that cells were here first. Reason being is a virus needs a cell to reproduce. So if there were no cells, the virus would have just chilled there and never been able to reproduce or do anything. So the viruses need a host, in other words, to become more populated. Okay, so we got our cells, we got our DNA that made up our cells. In some organisms at the time, it might have been RNA. But the second thing is, where did the viruses get their DNA from? Well, there are plasmids, which we talked about them in a previous chapter. That's the part of a bacterial DNA where we can insert genes of interest in order to use that DNA and have a complete set. There's also things called transposons, and these are just small mobile DNA segments. They can move around. So we think the viruses might have used both of these in order to get their DNA. Okay, so last thing for this whole chapter, guys, let's talk about how viruses actually, when they're in our bodies, how we get rid of them. All right, so that's our last thing. Now, when a virus gets into your body, its goal is to reproduce, and it also can do some damage. The first thing there talks about lysosomes releasing enzymes, and that can do damage. It can also produce some toxins, and that's what gives us our certain symptoms that we have when we have a virus. Now, antiviral drugs, if you guys have heard of antibiotics and stuff like that, antibiotics are things that can treat bacterial diseases. So if you have a bacterial disease, you go to the pharmacy, you get some antibiotics, and you're good in a few days. Now, antiviral drugs are a little bit different. Vaccines. So you guys know the big one now, obviously, the coronavirus vaccine. There's 3 different ones. There's the Pfizer, the Moderna, and then the Johnson & Johnson one that just recently came out. But these vaccines are used to try to not prevent us from getting the virus, but the goal of these antiviral drugs is actually going to be to stop the virus from replicating. So if we can stop the virus from replicating, then we can pretty much cure ourselves of the virus. So the virus isn't going to become plentiful in our bodies. It's not going to circulate to other areas. It pretty much just stops. So that is our goal. Our goal, especially like the coronavirus, is to stop the coronavirus from going to different parts of our body. Okay, so here's the West Nile virus, if you guys have heard of that one. That was back, 20-something years ago now. But West Nile virus, once that came, and that was carried like by mosquitoes, once that came over, it spread very, very quickly. And then Zika was even more recent than that. The Zika virus, it was like, oh, watch the mosquitoes, you might get Zika. All right. Now, let's say hypothetically, you get a virus and it infects a cell, one single cell. If that cell cannot go normal cell division, guess what? The virus really isn't going to do anything. If the cell is in like G0, if you remember that from interphase, and the cell is not reproducing, doesn't really matter. That virus is not going to spread to other cells too much. All right. All right. So just some other things. We got H1N1. If you remember that one, that was just a little over 10 years ago. But that was in Mexico and the United States. We had a big pandemic then, but it wasn't like as big as the coronavirus one. It was pretty much flu-like symptoms, just like the coronavirus, but not as severe. All right. Almost done, guys. So here are some things that contribute to viruses emerging. And you guys can read all of them, but I just want to comment on the first one and the third one because they're pretty much related to one another. So the mutation of existing viruses, which is especially high in RNA viruses, again, because we said RNA polymerase doesn't have that proofreading thing. And then the last one, spread of existing viruses from animal populations. About 3/4 of new human diseases originate this way. So if we combine these two together, so we have an animal virus that might make its way into humans. That's how the coronavirus happened. And when we have mutations in that virus, now we're getting new strains of that virus. Or we could even say it started off like, I don't know, we'll just say a bat. And the bat cell was so far off from our cell that the proteins on the outside of the virus couldn't infect our cells. Well, if there's a mutation, now it can get into our cells. And it would be a random mutation. It's nothing like, oh my goodness, I know I got to get into this human cell, let me mutate it. It's not like that. We have millions upon millions upon millions of viruses that get made every day, and if one of those decide that, hey, it's going to mutate, that one mutation in the protein can cause it to get into humans. All right, I think we got 2 more slides here, and then we're done. I'm just leaving the lights out, guys. Sorry, it's going to be dark. So just to talk about plant viruses really quickly. So plant viruses, they're pretty much the same as animal viruses. There's more RNA viruses for plants than there are DNA. And then the last thing, so we got to go over horizontal and vertical transmission. So these are with plant viruses. So they're spread 2 different ways. Infection from an external source of virus is called horizontal transmission. So that's the first one. Second one, the virus can be passed from parent to offspring. That is vertical. So if you just get the virus, like if it's a plant and it gets the virus, it's like, maybe in the wind or whatever, that's horizontal transmission. If the plant inherits the virus, that's vertical transmission. All right, guys, so that's it with chapter 17 on viruses. I told you guys it was a real short chapter. I got a video for you today on coronavirus. Watch that and finish your study guide. Have a good rest of your day.